Good morning, dear students. Today we will discuss an important and neat topic, anatomy of flowering plants. What do you mean by anatomy? Anatomy, it is the study of gross internal structure of a plant organ. The father of plant anatomy, Nehemiah Grew. He is considered as the father of plant anatomy. So the first thing we will discuss about the tissues. Tissues are a group of similar cells which are alike in origin and perform the same function. So group of similar cells or sometimes dissimilar cells, they may perform the same functions. Okay. So tissues are classified into two main criteria. One is simple tissue, another one is complex tissue. Simple tissues are similar in nature, complex tissues are dissimilar in nature. When it comes to broad classification of plant tissues, it is classified into two. One is the meristematic tissue, another one is permanent tissues. Meristematic tissues, a group of immature cells, show active cell division in response to some stimuli. The stimuli, it may be a internal stimuli or it may be an external. Based on that stimuli, the cells undergo continuous cell division. When come to the characteristics of the meristematic tissues, so it is a thin-walled, isodiametric in shape without intercellular space. It have a very dense cytoplasm with a prominent nucleus. A very good active nucleus is present in meristematic tissue. Vacuoles are absent. Endoplasmic reticulum, mitochondria are underdeveloped. Ergastic substance are also absent. But certain exceptions are there. If you take a vascular cambium, the vascular cambium, it is not an isodiametric shape, it is fusiform in shape, vacuolated one, and the walls are not thin, it is thick. Okay, and ergastic substance may be present. It forms two types of cells. Okay, the meristematic cells, it forms two types of cells. One is initiating cells and permanent cells. So initiating cells, it always remain in immature state. So the initiating cells always undergo continuous cell division. And the permanent cells, so the meristematic cells that become differentiated and form permanent cells. So when come to classification of meristems, based on the position, the meristems are classified into three main types. Apical meristem, intercalary meristem and lateral meristems. Apical meristems are present in the shoot and root region. So in the shoot region as well as in the root region, you may find the apical meristem. Okay, so this meristem is responsible for the plant growth. So that's why apical meristem, we call it as a growing point. Okay, this is, we call it as growing point. So when come to intercalary meristem, in intercalary meristem, so this is found in the nodal region. In between the nodal region, the intercalary meristems are present. So how this intercalary meristems are formed means, from the apical meristem, some of the meristematic cells, they will be separated or detached. So that intercalary meristem, that separated cell act as an intercalary meristem and start elongation of internodes. Okay. The third one is a lateral meristem. So lateral meristems are formed from the lateral meristems are actually formed from the permanent cells, permanent tissue. So the permanent tissue undergo differentiation, redifferentiation process and form cambium. So from that, what will happen? Plants will be, the phloem and xylem will be developed. So the example for lateral meristems are vascular cambium, cork cambium. So cork cambium is none but phloem. So other classification, origin, based on origin, primary meristem and secondary meristem. So primary meristems are embryonic meristems, promeristem we call it as. Okay, so from the embryonic stage, this meristems is present. Okay, apical meristems comes under primary meristem. Then come to secondary meristem. Secondary meristems are originated from the permanent cell. are from primary cells, okay, permanent cells. So if you take secondary meristem, especially core cambium and interfascicular cambium, everything comes under secondary meristem. Come to functions of based on the function, meristems has been classified into protoderm, procambium and ground meristem. Okay, so protoderm that gives epidermal tissue system. If you take this here, the protoderm, that will give the epidermal tissue system. Then procambium. So from the procambium, what will happen? The stellar evolution, steels will be formed. Okay. Then from the ground meristem, the cortical region will be developed. Okay. Next, a plan of cell division. If you take the plan of cell division, based on this, three main types of meristems has been classified. One is the mass meristem, another one is a rib meristem or file meristem, then plate meristem. Mass meristem, that will divide continuously, periclinally and anticlinally, and produce some tissues like endosperms, young embryos and sporangium also. If you take a rib or file meristem, that will divide anticlinally, but the division is in only one plan. Division is in only one plan. Okay, so very good example for rib meristem is development of cortex and pith. So cortex and pith is a rib meristem. Okay, so kindly note it down, dear students. Then plate meristem. If you take a plate meristem, plate meristems also divide anticlinally, but the plan of division is two sides. Okay, so very good example for plate meristem is epidermal development. Next, theory is explaining the apical meristem. So several theories which explains the apical cell apical meristem. The first theory is apical cell theory. So in apical cell theory, what will happen? The single cell is responsible for the formation of an entire plant. So let us take an example: algae, Bryophyta and Pteridophyta. The entire plant is developed from the single apical cells. Okay. So but this theory is not applicable for Phanerogon. Now this theory is proposed by Nageli. The second theory is histogen theory. Histogen theory is proposed by Hanstein. In this theory, three distinct zones is present in the meristem. One is a dermatogen, another one is a peribulum, another one is a plerome. If you take dermatogen, so dermatogen region, this will form the epidermal system. Then the peribulum region, this will form the cortex, and the plerome region, this will produce the steel. Okay, so this theory is only applicable for the root apex. This is not true for shoot apex. Next, when come to tunica corpus theory. In tunica corpus theory, this theory is proposed by Smith. So here the two distinct meristematic regions is present. One is a tunica, another one is a corpus. Okay, if you take a tunica is an outermost, divide only anticlinally, and enlarge is the surface area. Next, corpus is the innermost. From that, the remaining tissues will be formed. So this theory is applicable for the shoot apex. The another one is quiescent center concept. In quiescent center concept, if you take a root tip, there is some inactive zone. So that zone we call it as quiescent center zone. Okay, so in that the nucleus is not a prominent one. The very little nuclear cytoplasm is present in that cell. Okay, so when the meristem will be damaged, so this will become active. Okay, another one thing, meristem is a calyptrogen. From that calyptrogen, what will happen? The root caps will be developed. Okay. Next, when come to permanent tissue. So permanent tissue, it is composed of mature cells. Permanent tissue is composed of mature cells. The immature cells that become permanent, differentiated into form of permanent cells. Okay, it have a very good shape, size and function. So once if the cells become permanent, this loses the power of division. Okay, temporarily or permanently. Okay, some on the basis of development, permanent tissues are classified into two main types. One is primary permanent tissue, another one is secondary permanent tissue. If you take primary permanent tissue, the tissue which is developed from the apical meristem, that is comes under primary permanent tissue. and the tissue which is developed from the lateral meristems comes under secondary permanent tissue. Okay. Then simple tissues based on the cell constituent, that means the cell shape and function. The primary permanent meristems are classified into simple tissues, complex tissue and special tissue. Simple tissue means it have similar cells performing the same function. So simple tissues are three: parenchyma, collenchyma and sclerenchyma. Complex tissue are dissimilar cells but doing the same function. Xylem tissues, phloem tissue. Next, we come to specialized tissue. Some tissues and do some specialized work. Okay, so that things comes and that tissues comes under specialized tissue. So two types of specialized tissue we will discuss. One is lactiferous tissue, another one is the glandular tissue. So simple permanent tissue. Simple permanent tissue are a group of similar cells that will perform the same function. Very good, important simple permanent tissue is parenchyma. It is the most primitive fundamental tissues. The cell wall, it is very thin, okay, cellulosic in nature. Where if you take a parenchyma, one exception is there: wood parenchyma. Wood parenchyma, the cell wall, it is not thin, it is little thick, okay, and slightly lignified too. Then come to the shapes of parenchyma. Different shapes of parenchymas are there: polygonal shape, oval shape, spherical shape. So different shapes of parenchymas there. Okay, so parenchyma have intercellular space. It is a living one with a very good active nucleus. Then come to various functions of parenchyma. So various functions of parenchyma: erenchyma, storage parenchyma, stellate parenchyma, chlorenchyma, prosenchyma. So erenchyma are a special parenchyma. In that what happened, the air will be occupied in the intercellular space. So due to that, what will happen, the plant body possesses air. So due to that, what will happen, that have the ability to float in the water. So it get the floating buoyancy. So this erenchyma is present only in the plant which is floating. So example, nymphaeum. Okay, next is storage parenchyma. If you take a storage parenchyma, some parenchyma store food material and some it will store water. So for mainly in the form of stem tubers, root tubers, like that. Some parenchyma it will store the food. Some parenchyma stores water. So let us take a xerophytic plant, succulent plant that stores water. Okay, that is also comes under storage parenchyma. Some parenchyma are stellate in shape, star shaped. That's it. This kind of parenchyma is found in the petioles of banana and canna. Then chlorenchyma. So chlorenchyma is a kind of parenchyma in that chlorophylls are present. So that means this parenchyma involved in photosynthesis process. So this chlorenchyma is well differentiated into phallicide and spongy parenchyma. And prosenchyma. Prosenchyma or parenchyma, it is an elongated pointed one. This will provide a mechanical support. This will provide mechanical support. Okay, the next very important one is idioblast. So idioblasts are cells, parenchyma cells, which stores the resin, tannin, crystals of calcium carbonate and calcium oxalate. That means a waste product has been stored in the cells. So this kind of parenchyma cells, we call it as idioblast. Then another important function of parenchyma is protection. If you take an epidermis, epidermis is a parenchymatous in nature. This will give a protection to the entire plant system. And meristematic activity. So parenchyma have meristematic activity. Even though it is a permanent, sometimes the permanent this will be de-differentiated and re-differentiated and act as a meristematic tissues. Wound healing properties it shows. And regeneration property it have. Next when come to collenchyma. Collenchyma are simple. Collenchyma are also a simple permanent tissue which is a living one, and it give somewhat mechanical support to the tissue. Okay, so occurrence, if you take where it is found, means it is found in the hypodermal position of dicot stem. So kindly note this hypodermal position of the dicot stem. So 2 to 3 layer of hypodermal cells have been made up of collenchyma. And if you take a collenchyma is totally absent in monocot as well as in the roots. Next, if you take a shape, it is a polygonal shape. Cell have a very active and vacuolated protoplast. It have then if you take a cell wall, it is composed of cellulose, hemicellulose, and pectin. Okay, when come to function of this collenchyma, I already previously said that it is a mechanical in strength. It gives flexibility and elasticity to the tissue, and collenchyma it also perform the photosynthesis process. When means if it have chloroplast. Then based on the cell wall, the collenchyma is classified into three main types: angular collenchyma, lamellar collenchyma, and lacunate collenchyma. In angular collenchyma, cells are irregularly arranged. The corners of the cells have been thickened, and in this region there is no intercellular space are present. When come to lamellar collenchyma, the lamellar collenchyma arranged in the layers. Okay, and in a lacunate collenchyma is also thickened cell wall, but in this corners we may found some intercellular space. The next sclerenchyma. If you take a sclerenchyma is a simple dead mechanical tissue without protoplast. It is a simple dead mechanical tissue without protoplast. Okay, so then why it is present means this will give the mechanical strength to the plant. Okay, so if you take a cell wall, it is lignified one, and then shapes. So based on the shape and the length of the sclerenchyma, two main types of sclerenchyma has been classified. One is the fibers, another one is scleroids. Fibers, if you take a fibers, so fibers are elongated with pointed end. So both the ends are pointed, and the lumen. If you take the lumen are also not wide, it's narrow. Okay, so this have some simple pits in its body. Then if you take occurrence, where it is found, fibers are highly found in the hypodermis, pericycle, cortex, pith. If you take a bundle sheet of monocot bundle sheet, it is made up of fibers. Okay, and found in xylem. If it is associated with xylem means that we call it xylem fiber. If it is associated with the phloem means that we call it as phloem fiber. So based on the position, the fibers has been classified into wood fiber, blast fiber, and surface fibers. So wood fiber, it is associated with the secondary xylem. Okay, so in the stem region, especially in the stellar region, secondary xylem, if it is associated, that we call the wood fiber. Then if it is found in the phloem region, okay, that we call it as bast fiber. If the fiber is present in the phloem, that is bast. If it is present in the xylem, means it is wood fiber. Then some fibers are present in the surface of the plant organs, so that we call it as surface fiber. Very good example of the surface fiber is cotton fiber. So especially if you take a seed, the testa of the seed, it will start producing the fibers, that we call the surface fiber. Another one is coconut. If you take a coconut, have three distinct regions. One is endocarp, mesocarp, and epicarp. So in the mesocarp region, it have fibers. So these fibers have a very great commercial value. Then come to sclerites. Sclerites are slightly elongated dead cells. Okay, slightly elongated dead cells. It's not too much elongated, slightly elongated. It also have a lignified cell wall. It also have a lignified cell wall. The lumen is very much reduced here. Okay, so it also contains simple and bordered pits. So if you take a fiber, it contains only simple pit. It can also give mechanical strength, especially stone cells. That means the sclerites are found in the seed coats and endocarp. That's why endocarp is so strong. Types of sclerites: brachysclerites, macrosclerites, osteosclerites, asterosclerites, and trichosclerites. Brachysclerite are isodiametric in nature. This is found in the soft part, fleshy portion of the plant. Okay, so if you take pulps of Pyrus, in this you may find brachyscleroid. Macroscleroid are somewhat larger, elongated in shape. Okay, almost it's rod shaped, found in the seed coat, especially if you take a pea, you may find macroscleroid. Then osteoscleroid, name itself, it shows it appears a bone cell shape. Okay, then it is a rod shaped with a dilated end. They occur in leaves and seed coats. Then asteroscleroid, the name it appears star shaped. Okay, then trichoscleroid, it appears like a trichome structure. Okay, it may be branched or unbranched. The next, complex tissue. Complex tissue are dissimilar cells but performing the same function. Okay, so that will work together as a unit and do a same work. So two types of complex tissues, one is xylem tissue, another one is phloem tissue. Xylem, the main important function of xylem is water and mineral conduction. Okay, if you take this xylem structure, it has four different cells: tracheids, xylem fiber, vessels, and parenchyma. Xylem parenchyma. So in this four, xylem parenchyma is a living one. Then tracheids. So if you take a tracheids, this is the most primitive type of conducting tissues present in all the group of vascular plants. So almost this is found in all the vascular plants: pteridophyte, gymnosperm, and angiosperms. Okay, so if you take the shape of the tracheids, it has a tapered end. The end is a tapered, imperforated end, and it's a dead cell. It is thickened with lignin. So it has the cell wall deposition have different shape. It shows annular, spiral, reticulate, scleriform, and even piton. So according to the deposition of cell wall, different forms of tracheids will be formed. So it have some bordered pits. So with the help of this bordered pit only, the water conductions will be takes place from one trachea to the another trachea. Okay. Next, vessels. Vessels are long tube-like structures. Vessels are long tube-like structures. Okay, so it is a very advanced form. It is a very advanced form of conducting tissue, especially found in the angiosperm. Okay, so their walls are end with the perforated plates. Okay, some perforated plates are there through which the water conduction will be go to the another one vessel to the another vessel. So vessels are absent in certain dicots. So example, Drymis, Trichodendron, and Tetracentrons. And vessels are present. Okay, surprisingly, this in pteridophytes like Selaginella, Equisetum, and Marsilia, we may found vessels. And similarly, we may found in some gymnosperms also, Epidra, Valvicea, and in Netum also. Next, xylem fibers. If you take a xylem fibers, the sclerenchyma fibers is associated with xylem, that we call it as xylem fibers. Okay, if the xylem fiber is present, that will provide mechanical strength. Okay, then xylem parenchyma, a parenchyma which is associated with the xylem, that we call it as xylem parenchyma. So mostly in xylem, wood parenchyma is present. So it is a living, but the cell wall is somewhat thick. Okay, and it is slightly lignified. When come to phloem, phloem is the food conducting heterogeneous complex. Okay, mainly conduct the food in bidirectional. However, when come to xylem it is a unidirectional flow of food water conduction. But in phloem it is a bidirectional one. Phloem composed of sieve elements, companion cells, phloem fiber, and phloem parenchyma. Okay. First, we will discuss about sieve elements. Sieve elements are two: sieve cells and sieve tubes. Okay, sieve elements, the main important function is conducting of food materials. Okay, from source to sink. So sieve elements are very primitive conducting elements found only in pteridophytes and gymnosperms. Okay, And the important point you have to note that the sieve cells are not accompanied or associated with companion cells. Okay, sieve cells almost the shape remaining thing is resembles with the tracheids. Okay, it is analogous to the tracheids. Okay, so if you take a sieve cells have little minute pores, several small minute pores are there. That pores we call it as sieve area. Then come to sieve tubes. If you take this structure here, yes, this is a sieve tube. Okay, so it is an elongated tube-like structure. So this structure is found only in angiosperms. Okay, and perforated. Okay, so both the ends are perforated. So this end plate is present. That plate we call it as sieve plate. Okay, and it has some lining containing. That lining we call it as cytoplasmic lining. It has some special P protein, that means a phloem protein, that we call it as a slime body. Upon maturation, what will happen? The nucleus will be totally get disintegrated. That means absence of nucleus during immature. And sometime what will happen? Callose is a substance which will block this sieve plates. So what will happen? The nerve passage will be taken place. So when the dilute solution is coming, means automatically the callose will be get dissolved and the plug will be removed. So here the sieve tubes are always associated with the companion cell. Okay. So first, next we will discuss about the companion cell. Companion cells are a special kind of parenchyma cells. Okay, it is a kind of a parenchyma cells associated with sieve tubes. And one more thing, it's the origin of this companion cell is the same as sieve tubes, because both the companion cell and sieve tubes are originate from a single origin. So after the origin, what will happen? The meristematic cell divide and form sister cells. Okay, one form companion cell, another form sieve tubes. So then why companion cell is always associated with sieve tubes means the main important function of this companion cell, this will load the sucrose into the sieve elements. Okay. Then the companion cells are only present in angiosperm. The companion cells are only present in angiosperm. It is absent in the remaining pteridophytes, gymnosperm. But in gymnosperm, instead of companion cells, albuminous cells are present, associated with sieve elements. Okay. The next is a phloem fiber. If the fiber associated with the phloem, that we call it as a phloem fiber, it is a dead lignified one, mostly sclerenchymatous in nature. So phloem fibers, otherwise we call it as a bast fiber, very high useful in commercial industries. Okay. So pteridophyte, absence of phloem fiber. And if you take a phloem parenchyma, the parenchyma present in the phloem is a phloem parenchyma, and the primary two types of parenchyma is present. One is axial, another one is a ray parenchyma. So both the parenchyma do the conduction of food material. Okay, so if you take a ray parenchyma, that will helps in the radial conduction of food material. So phloem parenchyma is absent in monocot. Kindly make it notes that absence in monocot, present in pteridophytes and gymnosperm and dicot. Then special tissues. If you take a special tissues, a group of cells which do a special functions. That means what kind of special functions? Either this will release, or either this will secrete or excrete or metabolic end products. Okay, so these things comes under secretory tissues. Okay, or special tissues. So what are the secreting substance which is produced by the plant? Means is, plants mostly produce enzymes, nectars, mucilage, oils, all other things. Okay, when come to the types of specialized tissues, the two main types of specialized tissues are recognized. One is a glandular tissue, another one is a lactiferous tissue. If you take a glandular tissue, they will secrete out the material once it is formed. Okay, immediately after formation, it will secrete the secreting substance. But when come to the lactiferous tissue, the product it will be formed, but it is stored in the vacuoles. So after the cell will be get lysed, then only the product will be released out. Okay, so glandular tissue and its functions. Digestive glands are present in the plant, especially insectivorous plant, as a extra secretory one, and produce numerous of digestive glands, digestive enzymes, so through which when the insect will be trapped, so the nitrogen content will be absorbed by the plant after complete digestion. Next one come to nectaries. Nectaries, flower possess nectars, that means a sweet sugary content which is taken up by the insects during the pollination. Okay, so nectars have two types. One is floral nectars, another is extra floral nectars. If you take a floral nectars, it is found in the floral part of an flower, especially calyx, corolla, androecium and gynoecium. If it is present, that is a floral nectars. If the nectars are present in the extra floral, especially petiole, peduncle, in that region, if it is present, that is extra floral nectars. Osmospores, okay, flower, some scented fragment will be emitted due to the presence of this kind of glandular tissue. So it will secrete fragments, terpenoids and essential oils. Next are hydrothodes. Under root pressure, some plant, especially 2 to 3 centimeter height, such plant that will produce a droplet from the leaf margin. So okay, in the hydrothodes. Okay, so that kind of glands we call it as hydrothode glands. Okay, that discharge water and minerals by the process called as guttation. Next one is the lactiferous tissue. If you take a lactiferous tissue, lactex will be formed. If you take a lactex, what is lactex? Emulsion of protein, sugars, alkaloids, enzymes, watery fluid and rubbers, everything is present. Okay, so latex or white in color somewhat, sometimes it's yellowish in color, and even colorless latex is also produced by some plants. So mostly if you take a Euphorbiaceae family, latex will be produced. Okay, so latex. The next tissue system, 3 main types of tissue systems: epidermal tissue system, ground tissue system and vascular tissue system. Epidermal tissue system, if you take epidermal tissue system, it is produced or originated from protoderm, ground tissue peribulum and vascular tissue system from plerome. Okay, so epidermal tissue system, outermost a single layer coated with cuticle almost, derived from protoderm, parenchymatous in nature. So epidermal tissue system have epidermis, stomata, trichomes, hairs. The main important function of epidermal tissue system, that will protect the plant from the external environment. Gaseous exchange will be done. Secretion and transpiration control will be done by the epidermal tissue system, because opening and closing of stomata for transpiration is with the help of epidermal tissue system will be done. Next, if you take epidermis, epidermis doesn't possess any chloroplast, but however some cells possess chloroplast. For example, guard cells have chloroplast. Okay, and another one exception is sciophytes. If you take a sciophytes, it's a shade loving hydrophytes. Okay, in this you may find chloroplast. Next, bulliform cells. Monocot leaves under certain stress condition or in a dry condition, it will what will happen, the motor cells that become a roll up. Okay, that cells we call it as bulliform cells. It is also found in the epidermis. Next, cystolith. Cystolith, it's found in the ficus, the epidermal portion of ficus. It may found cystolith. It is filled with calcium carbonate. Next, stomata, epidermal appendages. One of the very important one is stomata, minute pores which have guard cells, 2 guard cells. Based on the guard cells, if you take monocot, dicot, different types of guard cells. If you take a monocot, it's a dumbbell shape. In a dicot, almost it is bean shape. Okay. In addition to this guard cells, so if you take a guard cells, I am already told that it possesses chloroplast, so photosynthetic process will be done. So to that, the other epidermal cells is subsidiary cell. So subsidiary cells is also a very specialized cell which also help in the transpiration, opening and closing of stomata. Next, sunken stomata. If you take a sunken stomata is a characteristic property of a xerophytic plant, especially if you take a cycas and nerium. So this is a sunken stomata region. Then adaptation. So if you take a position of stomata where the stomata is present high and low. Okay, so if you take a dicot plant, in the mostly the stomata are found in the lower epidermis. If you take a monocot plant, both the region you may found a stomata. If you take hydrophytes, only in the upper epidermis you may found the stomata. If you take submerged hydrophytes, there is no even stomata. Okay, then trichomes. So trichomes may be branched or unbranched. So here, if you take this diagram, it is a structure of trichome. This is a glandular one, especially found in the some insectivorous plant. This kind of trichomes is present, and the main important function of this trichomes, this will involve in the dispersal of seeds and fruits. The next is the root hairs. The important, everybody will know, increase the absorption of water and minerals. Okay. Next we will discuss about ground tissue system. If you take that, where actually the ground tissue system is present. Epidermal system is the outermost, now we discussed. Next, ground tissue system, this is a present between epidermis and the vascular system, that is called as ground tissue system. It is formed from the ground meristem, derived from the ground meristem. Okay, so if you take ground tissue system, distinct tissues are there: hypodermis, cortex, endodermis, pericycle, pith and medulla. Hypodermis, almost if you take dicot stem, it is a collenchymatous in nature, monocot stem, sclerenchymatous nature. So the layer which is next to the epidermis, that is hypodermis. Function protective and mechanical. Cortex, is several layered, mostly parenchymatous, have the food storage property in it. It do photosynthesis sometime, and even if you take a hydrophytes, aerial parenchyma cells are present. Due to that, the floating buoyancy it will get. Endodermis, if you take endodermis are single layer, barrel shaped, okay, which contains starch. If the starch is present in the endodermis, that we call it as a starch sheet. Endodermis, another one important we have to discuss, endodermis possesses Casparian strips. The Casparian strip, it's none but it's made up of lignin, suberin and cutin. Okay, so endodermis is completely stripped with this lignin, suberin and cutin, but there are certain cells are there where this lignin, suberin and cutin are not present. That cells we call it as a passage cell. So through the passage cell only, the absorbed water will be transported from the cortex region and to the protoxylem will be done. Okay. Then pericycle. We have to know more about pericycle because lateral roots are always originated from the pericycle. Kindly note, dear students, the lateral roots are originated only from the pericycle. If you take a pericycle, it is parenchymatous mostly, and sometimes sclerenchymatous also. If you take similax root, it is sclerenchymatous. And parasitic plant, it doesn't possess this pericycle. Okay, because in parasitic plant lateral roots will be not originated. The next, pith. If you take a pith, is the central portion. Parenchymatous, it may some function as a storage function. And if you take a medulla or pith ray, medullary ray, pith ray, so this will connect pericycle and pith, especially through this translocation of food material. So these are all those things comes under ground tissue. The next, vascular tissue system. if you take a vascular tissue system, the central cylinder steel, okay, which is composed of xylem and phloem, primary xylem and primary phloem, and even some in dicot cambium is present and absent in monocot. So, all the vascular bundle that constitute the vascular tissue system. If you take a primary xylem are two. One is protoxylem, another one is metaxylem. Protoxylem is the first formed one, metaxylem is the late formed one. Okay, so if you take a protoxylem, tracheids, vessels, parenchyma are present. Okay, tracheids, vessels, parenchyma are present. However, which one is absent? Xylem fibers are absent in the protoxylem. Okay. So, based on the position of protoxylem and metaxylem condition, three main types of vascular bundles have termed. One is a exarch condition, endarch condition, and mesarch condition. If you take a exarch condition, the protoxylem, it is pointed towards the periphery of the steel. Okay, it is always pointed away from the steel. That means towards, away. Then endarch, it is all the protoxylem, it is pointed not away to the through the center. Okay. Then mesarch, in mesarch what happened? The metaxylem, it is surrounds the protoxylem. Then primary phloem. So, what has we discussed in primary xylem, protoxylem, metaxylem? Here also protophlloem and metaphlloem, first formed one, late formed one. So, if you take a cambium, the next very important one we have to discuss about a cambium. Cambium is seen in dicot and gymnosperm. Okay, dicot plant and gymnosperm. However, it is absent in monocot. That's why in monocot secondary growth will not takes place, and in dicot and gymnosperm secondary growth occurs due to the presence of this cambium. So, cambium have two types. One is a fusiform initial, another one is a ray initial. Fusiform cells are elongated, tapered end, and the ray initials are isodiametric in nature. So, cambium, two types of cambium. One is a fascicular cambium, another one is interfascicular cambium. So, fascicular cambium, that is the cambium which is present in between the xylem and phloem in a vascular bundle, that is a fascicular cambium. That cambium is a primary meristem. So, during the time of secondary growth, what will happen? Some of the medullary cells that will be active and form cambium. That cambium is a interfascicular cambium. So, next, a type of vascular bundles. If you take a vascular bundle types, radial, in a radial vascular bundle, this is a radial vascular bundle. Okay, here, this one is a radial vascular bundle. In a radial vascular bundle, the xylem and phloem are arranged in a different radii. That means in alternative to each other. Okay, this is found in dicot root as well as in the monocot root. Then if you take a conjoint, okay, in conjoint, xylem and phloem are present in the same radius. Xylem and phloem are present in the same radius. So, collateral and bicollateral. If you take a collateral vascular bundle, in two types, one is open and closed. Okay, so here this is open vascular bundle. How it is open? Why we call it as open means due to the presence of cambium. Okay, so here this is closed because which one is absent? Cambium is not present here. Okay, that's why this is closed. Okay, so especially this closed vascular bundle is found in the monocot stem, and the open type is present in the dicot and gymnosperm stem. Then bicollateral vascular bundle, this is a very special. So, the xylem, it is surrounded by phloem on both the sides. Okay, so that two phloems are cambiums are present. Okay. So, very good example for this bicollateral vascular bundle is cucurbitaceae. Next, concentric. If you take a concentric, xylem and phloems are present in a circle. Okay, if xylem is surrounded by. If xylem is surrounded by phloem, that is amphicribal. If phloem is surrounded by xylem, means it is amphivasal. Okay, amphicribal and amphivasal. Then very important we will discuss now is dicot and monocot stem. The primary structure of dicot and monocot stem. If you take the dicot and monocot stem, most of the structures are similar, and somewhere you may found the dissimilarities. If you take a epidermis, here also it is uniseriate, here also it is uniseriate. Here epidermal hair is present. Okay, epidermal hair is present, but in come to monocot, epidermal hairs are not present here. Okay, without hairs. The next hypodermis. Here in monocot, the hypodermis, it is made up of sclerenchymatous in nature, but here it is collenchyma. I am already told that monocot, the dicot stem is a hypodermal region, 2 to 3 layers are there, that is collenchyma in nature. The next, if you take a cortex, so here a well differentiated cortex region is there, but here cortex is totally absent, because generally here we call it as ground tissue. No cortex is there, and in monocot the entire thing is ground tissue. Okay, so if you take a cortex, several layers are there. It's mostly made up of parenchyma, this region. Then endodermis. So here you may found endodermis, otherwise we call it as a starch sheet. Okay, due to the presence of starch, that we call it as a starch sheet. So here it is absent, no endodermis is there, but when come to here, one layered starch sheet is present. Okay. The next, when come to pericycle. In monocot, pericycle is absent. That's why lateral roots are not produced here. But when come to here, 1 to more layer of pericycle is present. Due to that, this is stem. So extremely apologize. Pericycle is absent here. If it is present here, it's made up of parenchyma and sclerenchymatous in cells. Next, medullary rays are there. Medullary ray here. Okay, this region is a medullary ray. It is present in dicot and it is absent in monocot. Then pith region, it is absent here, and here it is present. Next important thing we will discuss the vascular bundle. Vascular bundle, it is scattered here, because numerous of vascular bundles are scattered here and there in the tissue. But when come to here, the vascular bundles are always arranged in a ring. So here the vascular bundle are conjoined, collateral and closed. Why it is closed means there is no cambium here, and here this is conjoined, collateral and open, because phloem and xylem, in between that you may found the cambium. Next, it is oval shape. Okay, skull shaped, otherwise we call it as, but here it is a wedge shaped. Bundle sheet is present. So here this is a bundle sheet which is present here, which is almost sclerenchymatous in nature, but when come to here, bundle sheet is absent. Next, very important, we will discuss about the dicot and monocot root. If you take a dicot and monocot root, the most of the structures are similar. If you take epidermis, here and here it is a similar parenchymatous in nature, and a cortex is also similar. So several layers of cortex is here. Here also you may find several layers of cortex. But what happened here is, where in monocot root, sometime what will happen, the epidermis, this will be get shed out. That means this is fall off. So some of the layers, that means 2 to 3 hypodermal layer, this will become cutinized. That's it. Okay. So the cortex is made up of parenchyma. So and come to vascular bundle. So here the in dicot, the vascular bundle, it is different forms. Here the diagram, it's represented as a tetrarch condition, but some dicot, monarch, diarch, triarch vascular bundles are also there, up to hexarch. Okay. When come to here in monocot, the vascular bundle is polyarch. Okay, it is polyarch. Many xylems are present. Okay. And if you take the vascular bundle, it is a radial one, and the phloem and the xylem are present in alternative patches. Okay, so then the xylem and phloem is separated by a connective tissue. Here it is made up of a parenchymatous. So here this is a parenchyma one, and here it is a sclerenchymatous. Okay, so this is the difference between xylem and the monocot root and dicot root. If you take a pith, here the pith is absent, but when come to here the pith is present. So here the pith is present. Then if you take a dicot root and monocot root, some other differentiation we may have to note. One is the pericycle lateral roots. This will give very good. Okay, so from this only the lateral roots will be developed. Okay, apart from that, sometime phylogen also will be developed from the pericycle. In monocot root it's absent. Okay, so in the vascular, then we already discussed the conjunctive tissue, parenchymatous. Here it is a sclerenchymatous. Next cambium is present, that's why the secondary growth will be occur, and here it is absent, no secondary growth. So xylem usually tetrarch, and here it is usually polyarch in nature. Next, comparison of primary structure of dicot and monocot leaf. Okay, if you take a dicot and monocot leaf, the epidermis, the lower and upper epidermis. The lower epidermis and upper epidermis is a uniseriate epidermis. In a dicot, the stomata is present only in the lower epidermis here. Okay, but when come to here in upper epidermis as well as in the lower epidermis, you may found the stomata. Okay, that will mainly involve in the transpiration. And another thing, the next to the epidermis, here the cutin is present in the upper epidermis. However, here also the cutin is present in the upper epidermis. The next to the epidermis, you may found the parenchymatous region. Okay, mesophyll tissue is very well differentiated. Mesophyll, that is a phallicide parenchyma and spongy parenchyma. But when come to here, this is not differentiated. The mesophyll is not very well differentiated. Okay, phallicide parenchyma is somewhat elongated, and the spongy parenchyma is somewhat irregular in shape. Okay, next when come to the vascular bundle. If you take a vascular bundle sheet is present. Bundle sheet is present. Here also bundle sheet is present. Okay, so if you take a vascular bundle, some sclerenchymatous region is present in the both the side of the surface. Here it is collenchyma, somewhat collenchyma and parenchyma cells are present. Okay, next here mesar condition, xylem, phloem, no cambium is present. That's why in leaf no secondary growth will be there. And here in monocot, protoxylem, metaxylem, and are present. So this two is a metaxylem and protoxylem, and a phloem is in separately it is present. Okay, the main important function of leaf, everybody we know, photosynthesis. And one more point I have to remember, here it is this is we call it as bulliform cells, the main cells which involving in the rolling upon the stress condition. Okay, next. Take a stomata. I am already told that the leaf in dicot, the leaf is a dorsiventral, that means differentiated upper and lower, and monocot isobilateral leaves. Stomata usually more on the lower. Here in both the surface you may find stomata. Mesophyll tissue is well differentiated, phallicide parenchyma, spongy parenchyma. Here only the mesophyll is not differentiated. Bundle sheet is made up of parenchyma. Here also it is made up of parenchyma, but sometimes sclerenchyma bundle sheet is also present in the monocot. Bulliform cells are absent here, and here it is present. Okay. secondary xylem. If you take a secondary xylem, secondary growth. Secondary growth is none but when the primary plant body that undergo further growth by girth or the diameter of the plant if it is increased. So that we call it as secondary growth. Secondary growth occurs in dicot plant and gymnosperms, especially not reported in monocot, but some monocot also secondary growth is taking place. So why the secondary growth takes place? Due to the presence of lateral meristems. Okay, so lateral meristem, if you take a vascular two types of vascular lateral meristems, one is the vascular cambium, another one is corky cambium. If you take a vascular cambium, which is present already in between the xylem and phloem, okay, that is fascicular cambium, and the newly formed cambium from the medullary race, some of the medullary race that became meristematic, so that cambium we call it as interfascicular cambium. So this will form a ring-like structure, okay, that we will discuss now. Okay, and come to cork cambium. Cork cambium, sometime from pericycle or some of the cortical cells thus become meristematic and act as a cambium. So outer side that will produce a cork and the inner side that will produce a secondary cortex, okay, phellum and phallodum. So next we will discuss about the secondary growth, how it takes place in the dicot stem. Okay, so if I take a intercellular growth, so the growth how takes place in the vascular bundle. Then next we will discuss how the growth takes place in the cortex. So in dicot stem, the vascular bundle is usually, everybody we know, conjoint, collateral and open type, okay, arranged in the form of ring. So we already, I am told that fascicular cambium, interfascicular cambium, these two cambiums are there. The cambium which is present between xylem and phloem, which is a primary in origin, and from the medullary race, if some of the cambium that will arise, that is secondary in origin. So these two constitute and form a ring-like structure. Okay, so here this you may find clearly here, yeah, here. So here a ring-like structures will be formed due to the presence of vascular and fascicular and interfascicular cambium. So here this ring structure, this will start to divide from both the sides. Okay, so from one side this will start dividing phloem, another side this will start producing xylem. Okay, so the cambial ring cut off outside, okay, that will produce secondary phloem, and if it is cut off inner side, that will produce secondary xylem. So where the division is more means in the inside, the that means the secondary xylem side, this will be more, and thus what will happen, the growth of the stems will be taking place, the increase in the plant growth will be takes place. Okay, this secondary xylem is the major portion of the conducting of water. So based on the secondary xylem formed, okay, wood are classified into two main types. One is porous and another one is non-porous. Okay, if you take this angiospermic wood and this is gymnospermic wood. Okay, so angiospermic wood, you may find vessels, large sized vessels are present, but here gymnosperm vessels are absent. So that's why it is non-porous, and here it is porous. Okay, next we will discuss about annual rings. So take this image. So you may find several small circle, very differentiated alternative bands of light and dark rings are present. So this rings, what we call it as annual rings. Okay, the activities, how this annual rings are formed means, so due to the activity of cambium. So if there is a seasonal changes occur, so this rings will be formed. So according to annual ring, two types of rings will be formed. One is a spring ring, another one is autumn ring. Okay, that's we call it as a spring wood, another one is autumn wood. If you take a spring wood, how this spring wood will be formed? Okay, during this season, spring season, the cambial activity is more, thus the size of the cells will be more and the lumen is also more, and that's why the size is somewhat bigger. Okay, so if you take this image, so this is this light region is a spring wood, light in color, but when come here it is a darker. So another region is a darker, that is autumn wood. So why autumn wood is formed means in this season the activity of a cambium is slightly reduced, highly reduced, and the cells will be small in size, and the lumen is also become lesser. So that's why this appears a dark. Okay, so every year two types of rings will be produced. So this combined we call it as annual ring. So with the help of this annual rings we may calculate the age of the plant. Okay, so the study of this annual ring and we calculating the age of the plant, that we call it as a dendrochronology. Next we will discuss about heartwood and sapwood. Okay, so if you take this here, here if you take a heartwood and sapwood, in this wood two regions are there. So central region and the peripheral region, two regions. You may find that the two regions are slightly variation color. Okay, so central region is somewhat darker in color, it is harder and tougher. Okay, so that wood is we call it as a heartwood. Why this heartwood is formed means due to the formation of tyloses. What is this tyloses? Tyloses are a special structures. So here let us take, this is the xylem vessel. So this is a parenchyma cells. So this parenchyma cells that will start protruding, okay, inward to the xylem vessel through plasmodesmata and pits. So what will happen? The xylem will be completely blocked here. So here this is a balloon-like structures. This we call it as a tylosis, and this completely blocked. So due to that blockage, what will happen? This vessels will be deposited with the resins, tannins and gum, and no conduction of water will be takes place. So water conduction is stopped. So this wood is called as heartwood. When come to the softwood, how it is conducting activities. Okay, so it is a living part and it is a dead part, we call it as. Okay, and here tyloses are absent. So in this two, which part is more resistant to microorganisms and the environment? Okay, so here this heartwood is more resistant when compared to the sapwood. Next we will discuss about a cork cambium. To take a cork cambium, some cortex cells and pericycle, some of the pericycle cells that become meristem. Take activity. That shows some meristematic activity and start producing and dividing some layers of tissues. Okay, so the meristem cells we call it as cork cambium. So otherwise we call it as a phellogen. So the phellogen cut outward and inward. If it is cut, if the cork cambium is cut outward, that we call it as cork phellum. If it is cut inward, that we call it as secondary cortex. Okay, and the special structure is also present. So this combined cork cambium, cork and cortex. So this three we call it as periderm. In this secondary cortex and the cork cambium are living in condition. However, this cork is dead due to the deposition of suberin. And the main important function of this cork cambium is protective. Okay, this another thing important point we have to note that this cork cambium is extrastellar origin. And the next some important structure is lenticels. If you take, this is a lenticel structures, a small opening which is present in the bark. This is a lenticels. Okay, so here with the help lenticels, gaseous exchange, transpiration will be takes place. Somewhat loosening cells are arranged in this region. Okay, so here this is filled with some complementary cells. Next we will discuss about secondary growth in dicot plant. Okay, as if you take it's a radial vascular bundle, so here no cambium is present. So what will happen? Some of the cells which is present in the vascular bundle. Some of the cells which is present in the vascular bundle that became active and initiate like a cambium. Okay, the cells which is present below the phloem that became active and act as a cambium, and finally waving rings will be formed here. Okay, look clearly, you may find waving rings will be formed here. Okay, so and a stage will be developed, the complete ring will form, and one side that will start producing phloem, another side this will start producing xylem, as what we discussed previously in the dicot stem. Okay, and now we will discuss some of the previous NEET questions. Okay, the balloon structure shaped structure is called a tylosis. Okay, option A, the originate in the lumen of vessels. Option B, characterize the sap wood or extension of xylem parenchyma cells into the vessels, are linked to the ascent of saps through the xylem vessels. The correct option is this. The tylosis is a blockage which happened in the vessels. How the blockage is taking place? With the xylem parenchyma will be protruded inside. Okay, and the blockage will be happening. Okay, option is C.